The word inflammation is derived from a Latin word inflammatio, which means to set on fire. This is one of the reasons the title slide of my talk also had a fire in the background. The entire body is set on fire, something which I want to tell my student, that inflammatio means to set on fire. So what exactly is inflammation? Inflammation is a non-specific response to tissue injury. It can be caused by pathogen or it can be caused by physical trauma. That infection can also cause inflammation as well as physical trauma when you are hit by some sharp object or pointed object, or you are injured, road accident, there also you have inflammation. Acute inflammation is an immediate response, and this is a response of body to both injury as well as cell death. Now, what are the cardinal signs of inflammation? There are 4 cardinal signs of inflammation, and all 4 put together gives the 5th entity. The first cardinal sign of inflammation is redness, that is rubor. I have highlighted a rose red in color to suggest or make you remember that redness is rubor. Second is warmth or calor, sunlight, sun is hot, that is warmth or calor. Third is pain or dolor. Sometimes when you are injured, you writhe in pain, that is pain. And fourth, swelling, sojo, what you call in Gujarati, that is tumor. So 4 classical signs of inflammation are rubor, calor, dolor and tumor. All 4 put together leads to loss of function. This is called as altered function or functio lassing. Now, in the next part of our talk, we shall focus on the chemical mediators of acute inflammatory response. The chemical mediators of acute inflammatory response. What are the chemical mediators of acute inflammatory response? There are 3 mediators: selectin. These are the cell adhesion molecules on activated capillary endothelial cell, popular MCQ. Where do you find selectin during inflammation? Selectin is found on the activated capillary endothelial cell. It is on the capillary you find selectin. Integrin, quite opposite to it, is the adhesion receptor on the neutrophil. So again, students, please remember, I already talked in physical classroom. Integrin is on neutrophil and selectin is on capillary endothelial wall. Chemotaxins, the chemotactic factors released by injured cell. If you recollect, I threw the duster in one of the benches. One of your friend was very scared. Okay, and that was when I was explaining the chemotaxins and the chemotactic factor. The chemotactic factor released by the injured cells leads to the potent inflammatory response. Now, acute inflammatory process, there are various processes that occur. Some of the various processes are margination, dipedasis and extravasation. Okay, the extravasation, 3 aspects: margination, dipedasis and extravasation. What are the physiological events of acute inflammatory response? These are the classical physiological effects of acute inflammatory response. You have selectin integrin interaction. Again, where do you find selectin? Where do you find integrin? You have integrin on the neutrophil and the selectin on the capillary endothelial cells. So there you have the this is the neutrophil. You have on neutrophil you have the integrin. So neutrophil has integrin whereas the blue colored structure, this is the selectin on the endothelial cell. So, once the selectin and integrin locks itself, this neutrophil will stop rolling. Once it stops rolling, it will marginate towards the margins of the endothelial cell. Later on, it will squeeze through the capillary and it will reach the tissue space where the original injury has taken place. This is the diagram from your book, Prescott, and that leads to, then the phagocyte would come and the phagocyte would eat away the pathogen, that is phagocytosis, something which we covered in our very first lecture. So, these are the physiological events of acute inflammatory response. Now, the same event I will draw in a flow chart fashion. I think I covered during my physical classroom, just a sort of revision. You have injured tissue. Injured tissue will send chemical signal. This chemical signal will activate the inner lining of the endothelium. Once the inner lining of the endothelium is activated, the inner lining of the endothelium will express selectin, which is displayed within the capillary of activated endothelial cells. Selectin will lead to attachment and wandering neutrophil, they will get attached. The neutrophils will slow down, and once the neutrophils are slowed down, they will roll along the entire endothelium, that is the endothelial lining. Okay. Once the slowing down takes place, they will encounter activating signal. This will activate the integrin on neutrophil. The neutrophil and integrin will tightly attach to the selectin. In the next step, all are 1 mark short question, the next step onwards. The same question can be asked in short question. Someone asks you what is margination, you should pay attention to what I am speaking. The neutrophil integrin attach tightly to the selectin, and after attaching, the neutrophils will stick to the endothelium and they will stop rolling. This act of the neutrophil to stick to the endothelium and stop rolling, this is called as margination. So, margination is when it stops rolling. Neutrophil will stick to the endothelium and stop rolling is margination. The next term is diapedesis. Neutrophil, after it marginates, it will undergo dramatic change in shape. It will squeeze through the endothelial wall. This squeezing of the neutrophil through the endothelial wall is called as diapedesis. Another 1 mark question for examination. What is diapedesis? MCQ, short question, fill in the blank, whatever it is. This is diapedesis. And finally, the last term, extravasation. The neutrophil then goes to the interstitial tissue fluid. They will migrate to the site of injury. This is called as extravasation. What is extravasation? The migration of the neutrophil after margination and diapedesis is called as extravasation. Extravasation is the phenomenon by which the neutrophil will reach the tissue. Where the tissue damage has taken place, there the neutrophil will reach. This is called as extravasation. Now, this much part we have already covered in physical classroom. The next slide onwards, that is 50% of the lecture, is where we have to study. So, what tissue injury does? When you have an injury, what the tissue injury does to that particular site? So, what tissue injury does? The tissue injury will release a very important biochemical mediator, a kallikrein and other mediators. The tissue injury releases kallikrein. What tissue injury releases? Kallikrein, and along with other mediators. Now, short question for examination. What is this role of kallikrein? What this kallikrein does? 2 mark question. Kallikrein will increase the capillary dilation and blood flow. What is capillary dilation? The increase in diameter. This is the capillary, this is the circle, the center of the circle, this is radius, say 5 centimeter. Because of dilation, this radius increases from 5 centimeter to 10 centimeter, that is dilation. Vasodilation is increase in diameter. So, increase in capillary diameter, more capillary diameter, more blood flow. So, it increases blood flow. Along with the blood flow, it brings more and more antimicrobial factors and leukocytes. Why they are brought? They are brought to the site of tissue injury to kill the pathogen. To kill the pathogen, they are brought to the site of tissue injury. This will kill the pathogen or eliminate the pathogen. The next part of the talk, we will discuss the reactions at the site of injury. Lots of biochemical reactions take place at the site of tissue injury. We are all into that. First reaction is fibrin clot will restrict pathogen movement. Fibrin clot, students, you might have studied that it stops blood flow, but not only it stops blood flow, it also restricts the pathogen movement. In the meanwhile, the hug of death sets in, that is phagocytes will accumulate in the inflamed area and they in turn will destroy pathogen. The bone marrow in turn is stimulated and they in turn will release neutrophil and they will increase the granulocyte production. This I will talk in terms of analogy. You all have heard the Bollywood song Abhi to party shuru hui hai. So, whenever the injury sets in, a party starts at the site of injury, and that party which starts at the site of injury will recruit lots of neutrophil. Now, already neutrophils are recruited, we need more, still more neutrophils, still more granulocytes. So, the rate of granulocyte production increases. This is done by GM-CSF, not in your syllabus, but just general knowledge. GM, granulocyte monocyte colony stimulating factor, that is GM-CSF. This will increase the rate of granulocyte production. Now, bradykinin pathway, right side figure is from your book, left side is some extra thing which I have given to explain you. Imagine a splinter or a nail has come, your tissue is injured, your skin, you have the kallikrein, and this you have plasma kallikrein and tissue kallikrein, high molecular weight kallikreins and low molecular weight kallikreins. This kallikrein, use a highlighter, this kallikrein, if you see, is a long chain molecule. This kallikrein will be broken down into a short bradykinin, and bradykinin will have multiple effects on nerve cell, effects on blood vessel, effects on mast cell. All these effects we are going to study. So, what I will do, I will elaborate this diagram in 4 or 5 slides so that my students get a correct picture of what exactly is happening. You can see the within the capillary, the blood cells are floating. So, bradykinin will have effect on pain, it increases pain, it causes vasodilation, it increases vascular permeability, and it causes chemotaxis. So, we will take up each of them separately. This is from your book again, but I have simplified things. First step is inflammatory response, second step is increase in acidity, third step is all effects of bradykinin. So, the examiner might break the question and ask into multiple parts. Either it can be a 10-mark question or 5-mark question on write a short note on inflammation, acute inflammation, or several short questions such as explain the role of bradykinin in inflammation. What is the role of increased acidity in inflammation? Explain the important steps of inflammatory response. So, we will discuss all this. First step, inflammatory response. What happens in inflammatory response? In inflammatory response, we have release of inflammatory mediators. This is a cascade of events. All these cascade of events, few minutes before we discussed, these cascade of events will lead to development of signs of inflammation. So, first step is inflammation. After first step, what happens is there is an increase in extracellular fluid acidity, ECF acidity increases. acidic component of extracellular fluid will activate kallikrein. Now, kallikrein, once it is activated, the kallikrein, which is a long molecule, will undergo cleavage. This cleavage or breaking down will release bradykinin. Bradykinin will be released from a long precursor. So, someone asks you which is the lengthy molecule? The kallikrein is the lengthy molecule. Which is the short molecule? The bradykinin is the short molecule. So, what releases bradykinin? The kallikrein releases bradykinin. How it is released? Increase in acidity. What is the mechanism? Cleavage. What is cleavage? Breaking down. So, kallikrein is released, bradykinin is released from kallikrein, the long term precursor, by increase in extracellular fluid acidity. This release of bradykinin, now bradykinin will bind to the receptor on capillary wall and it has multiple effects. So, again this can be an exam question. Explain the multiple effects or explain the various effects of bradykinin. If this question is asked in examination, you have to separate out an answer. So, I am dividing the answer into 3 parts. Remember student, the question in examination can be what are the effects of bradykinin. So, I am dividing the answer into 3 parts. The first part is effect of bradykinin on capillary wall. So, effects of bradykinin on capillary wall, it will open the junction between the cells, right. So, the door will be opened. What will happen because of the door? It will allow the fluid and leukocytes to pass through. The capillary of the infected tissue will stimulate the production of prostaglandins and PGE2 and PGF2 alpha. Prostaglandins are mediators of pain and inflammation. That is why whenever you take anti-inflammatory drugs such as Combiflam, which contains brufen and paracetamol, these drugs will downregulate the prostaglandin pathway. PGE2 and PG2 alpha will promote tissue swelling, what you call in Gujarati as sojo, where in the infected area. So, someone asks you what is the effect of bradykinin on capillary wall? These are your points. Influx of fluid, the stimulation of prostaglandin production and tissue swelling. So, what are the next part is what are the effects of bradykinin on mast cell? First, diagrammatically, this is the mast cell degranulates. After degranulation, it releases histamine, tryptase, chymase, and all these mediators will have effect on the target cell. These are the anaphylactic shock or allergic reactions. So, bradykinin binds to the mast cell on the connective tissue. Where do you find mast cells? Mast cells are always found on the connective tissue. And mast cells are associated with very small blood vessels. The smallest blood vessel happens to be capillary. The largest blood vessel is that of artery, right, aorta of course. So, small capillaries rupture or break down, and this will activate the mast cell by influx of calcium ion. Whenever the calcium ion influx takes place, the mast cell degranulates, and mast cell degranulation leads to release of mediators such as histamine, tryptase, chymase, and this will all have effect on the target cell. So, degranulation release of preformed mediator such as histamine, and this is the reason whenever you suffer from allergy, we take antihistamine drugs such as cetirizine, levocetirizine and chromonil sodium. So, these are the effects of bradykinin on mast cell. Now, what are the effects of bradykinin on the nerves of the infected area? Obviously, whenever there is inflammation, we are going to get pain. So, how the pain is caused? So, damaged nerves release substance P. MCQ, what substance is released by damaged nerve? The answer is substance P. Substance P will bind to the mast cell, and again it will cause mast cell degranulation. So, this causes pain from the nerve endings. Now, we will see some of the mechanism by which the pathogen is eliminated. So, neutralization and elimination of offending pathogen. First of all, there is an increase in blood flow, there is a capillary dilation. Both increase in blood flow and capillary dilation will bring more blood into the area. More blood into the area means more antimicrobial factors, more leukocytes, and all these leukocytes or WBCs will destroy the pathogen. After the pathogen is destroyed, the dead host cells will release antimicrobial factors, and these antimicrobial factors in a reversible fashion will also destroy the pathogen. So, this is how the neutralization and elimination of offending pathogen takes place by acute inflammatory response. In the next part, we will still continue with neutralization and elimination of offending pathogen. In book, it is a paragraph, but I have simplified the steps so that you remember it nicely for exam. Again, the blood leaks into the tissue space. You cannot see external bleeding, but the blood, internal bleeding is always dangerous because the blood leaks into the tissue space. Because of the blood leaking into the tissue space, there is an increase in temperature. This causes calor, or whenever you get inflammation, the surface temperature increases because of this blood leakage. This increase in temperature stimulates the inflammatory response, and inflammatory response is very protective. It inhibits microbial growth. Someone asks you a question, how increase in body temperature or how fever acts as microbiostasis. This is your mechanism: blood leaking to tissue, increase in temperature, stimulation of inflammatory response. Inflammatory response will inhibit microbial growth. Next, I will discuss the fibrin clot and its mechanism. Again, there is a formation of fibrin clot. Because of the formation of fibrin clot, the spread of infection is limited and the infection or inflammation remains localized. Your entire body is not affected, only a selective part of the body is affected, that is called as acute or acute inflammation or localized inflammation. Systemic inflammation is when entire body is affected. COVID-19, why a patient dies is because of cytokine storm, and it is a acute inflammation where the systemic or the entire body is involved. Here of course, it remains localized, so elimination of pathogen takes place. Now, how the neutralization and elimination of the offending pathogen takes place? The phagocytes will go to the inflamed area, or it will translocate to the inflamed area, and there will phagocytose or eat up the pathogen. Additionally, there are chemicals, and these chemicals will stimulate the bone marrow to release neutrophils and increase the rate of granulocyte production. I just explained the role of GM-CSF, granulocyte monocyte colony stimulating factor, which increases the rate of granulocyte production. Again, further the neutralization and elimination of pathogen, you have phagocytes will collect in the inflamed area and they will phagocytose the pathogen. By phagocytosing the pathogen, they will release certain chemicals from the bone marrow, and these chemicals will increase the rate of hematopoiesis. They will release more neutrophils, and agents such as granulocyte monocyte colony stimulating factor will increase the rate of granulocyte production. More and more granulocytes will be produced. Now we move to the second part of our topic. Till this part was acute inflammation. Now short and sweet, chronic inflammation. Chronic inflammation, unlike acute inflammation, is a slow process. You don't die. You remain sick for an extended duration of time. It is a very slow process, and it involves formation of new connective tissues. They will cause permanent tissue damage. Unlike acute inflammation, chronic inflammation will cause permanent tissue damage. What happens in chronic inflammation? Dense infiltration of lymphocytes and macrophages take place at the site of inflammation. If you take a histological section of the injured area or inflamed area, you will find so many lymphocytes and macrophages which has accumulated at the site of inflammation. So, this is the characteristic microscopic observation of chronic inflammation, and chronic inflammation always has a granuloma. The granuloma will wall off the area, it will separate that area, or nakabandi it will create, wall off the area. It will form when a phagocytic cells cannot destroy the pathogen. This is chronic inflammation. I will explain it. This is the tissue, injured tissue. So, everywhere lots of phagocytes surround and they are releasing all inflammatory mediators, but they are not able to neutralize this pathogen. Instead of neutralizing the pathogen, they in turn destroy the tissue. This is called as the walling off the area and destroying the tissue. This is chronic inflammation. So, chronic inflammation, that is mycobacterium and leprosy, these are the diseases. Why these diseases should be treated? Because if you do not treat this disease, it will lead to the mycobacterium and leprosy will lead to certain inflammatory conditions. The cell walls have a very high lipid or wax content in both mycobacterium. Remember student, just for general knowledge, not for exam, mycobacterium it is mycolic acid, leprosy it is lipoarabinomannan. So, the cell walls have a very high lipid and wax content, and these cell walls are resistant to phagocytosis and intracellular killing. They will survive within the macrophage, that is why macrophage itself is a immune cell which kills, but these microorganisms survive within the macrophage, such as protozoans such as Leishmania, and there are certain bacteria also which produce toxins, and these bacteria causes tissue damage even after bacterial death. So, students, I am coming to my first point with which I started my lecture, that inflammation cannot just be because of tissue injury, it can be also because of infection. Viral infection causes inflammation, bacterial infection causes inflammation, and fungus also, aspergillosis also causes inflammation, right, Aspergillus niger. Now, what are the causative agents? Which are the agents that cause chronic inflammation? This is a 2-mark question that can be asked. Bacteria such as listeriosis, brucellosis, fungi such as histoplasmosis, coccidioidomyces, helminthic parasites such as schistosomiasis, protozoans, leishmaniasis, and large antigen antibody complexes. Best example is your rheumatoid arthritis, where antigen antibody complexes will accumulate in the, and it accumulates and causes stiffening, that is rheumatoid arthritis, osteoarthritis and rheumatoid arthritis. Finally, I will summarize my talk with that inflammation is a complex sequence of events which stimulates inflammatory response. Some of the classical features of inflammatory response are redness, swelling, production of heat, pain, and loss of function. Inflammation are of two types: acute inflammation, which is response to tissue damage and it limits infection. Another is chronic inflammation such as arthritis and wasting associated with certain cancers. Inflammation has 4 important steps. Tissue damage causes vasodilation, then you have permeable capillaries, which is this is called as edema. Finally, you have phagocytosis, and the phagocytes will destroy the pathogen, and these events include margination, dipedasis, and extravasation. Extravasation is the movement of the neutrophil from capillary space into the tissue space. Finally, thank you very much for your attention. A teacher is never successful without good students, and these are the list of some of the students whom I have taught. You have already seen them, most of them might be conducting your classes. You have your Megha madam, Manthan sir, Jayesh sir, Akash, Sonal as well as Chandni madam, all have been my students, and one of the reason behind portraying this to you is you also become disciplined and take your studies with a bit of passion.